Okay, welcome to Ask the Expert with Dr. Tarin Bigley. He's an MD, PhD. He's assistant professor in pediatric rheumatology and immunology. And he's coming to us from WashU in St. Louis, which is really becoming quite a powerhouse in the type 1 diabetes field. He's, as I said, he's an assistant professor, but he did get his MD, PhD training at the Medical College of Wisconsin, another excellent place. And let me just let someone in here. He's going to talk today about his work, which focuses on neonatal roseolovirus infection, which induces disruption of thymic tolerance and results in autoimmunity and autoantibodies, including, and surprisingly, including anti-insulin antibodies. Welcome, Dr. Bigley, and thanks for joining us.
Yeah, thanks so much for having me. I'm excited to talk about this research to this group. I think the initial studies maybe are somewhat type 1 diabetes adjacent, but have a lot of potential implications in terms of the phenotype we're seeing and also the impact on the immune system with type 1 diabetes. So I do not have any financial disclosures, but as a pediatric rheumatologist and immunologist with a background in virology research, I'm really interested in this idea that the difference between an autoimmune disease and an infectious disease is that in the latter, a pathogen has been identified. And we know that there are autoimmune diseases that are monogenic, that develop very early in life, at times before kids have seen a lot of infections. But most autoimmune diseases, there isn't a monogenic cause. They're heterogeneous in terms of presentation, age that they develop, and we think a lot about viruses and their potential role in autoimmunity. And so I'm going to discuss some of the findings that we recently published in JX Med on disruption of central tolerance and development of autoimmunity after a neonatal infection with murine roseolovirus. And then touch a little bit on some additional data, especially relating to type 1 diabetes. So the evidence for virus-induced autoimmune disease in much of the literature is epidemiologic or from case or cohort studies, and these are retrospective studies. They often assume this close temporal relationship between the onset of autoimmune disease and infection, but the pre-infection status is often unknown. These studies often lack causality and mechanism. I do want to give a shout out to a recent study on Epstein-Barr virus and multiple sclerosis, which probably has come as close as anyone has come to identifying a potentially causative role for a virus in an autoimmune disease.
Yeah, absolutely.
Yeah, but I've been studying a different herpes virus, roseolovirus, and these are species-specific beta herpes viruses. They have very high prevalence and are well adapted for chronic infection in their given host species. The human versions of these are HHV6 and HHV7. And in pediatrics, we see this all the time as roseola infantum. Kids get a high spike in fever followed by a rash. And we think of this as largely self-limiting, that these kids recover really well, and this is very common. It can be an opportunistic pathogen in immunosuppressed patients, but has been linked to various autoimmune conditions. The difficulty in studying this is that they have a very high prevalence. So early in life, seroprevalence approaches or is over 90%. And these are chronic infections that are latent throughout life with periodic reactivation. So causative studies are very difficult. And as I mentioned, these viruses are species specific. So studying the human roseola viruses in vivo is very challenging. And we therefore have been studying murine roseolovirus, which was sequenced in Wayne Yokoyama's lab here and found to be this virus, murine roseolovirus or MRV, was found to be highly related to the human roseoloviruses. Now, even in the wild, similar to people, this virus has a very high seroprevalence. But most mice with it seemed to be fine. What we did find is that after neonatal infection, mice develop thymic atrophy and T cell depletion. And you can see in this full plot here of the thymus, this is largely true of the CD4 positive thymocytes as well as peripheral T cells. But this is a transient phenotype. By the time we're weaning mice, we no longer see any of the CD4 T cell depletion. The thymus architecture has normalized. Any failure to gain weight has resolved. And this correlates with viral replication, shown here using viral RNA as a proxy, where we see after 2 weeks post-infection, we can no longer detect viral RNA in the thymus. And so we wanted to know if this had any long-term impact that maybe we weren't seeing grossly in the mice. And what we found surprisingly is that when we looked at 12 weeks post-infection at the stomach, we see this inflammatory infiltrate as well as disruption of the normal gland architecture. And this occurs at very high penetrance. You can see all of the mice, shown here for valve C mice, develop gastritis. We also see this for multiple different mouse strains, so this was not strain specific. But we did not see virus in the stomach at this time, and this is as opposed in opposition to the salivary gland, which we think is a reservoir of latency.
Mm.
So the other component of autoimmune gastritis is the development of autoantibodies. And when we take serum from infected mice that are 12 weeks old and stain tissue from stomachs, we see that compared to the mock or uninfected mice, there's positive staining from the serum from the MRV infected mice. And this is scored here. But we also see when we do an ELISA to a stomach specific antigen that these autoantibodies are present, really suggesting that this virus is inducing autoimmune gastritis after neonatal infection. So we wanted to look a little bit closer at whether or not this was dependent on the timing of infection and on viral replication. And what we did is we set up um, several different experiments where we either mock infected mice, and included a vehicle control, or infected mice along with an antiviral, in this case, ganciclovir. But we only did this for the first week of life. We also infected mice on day of life 7. Just showing here, the antiviral we use is highly effective. The runting that we typically see after MRV infection is rescued. We also see about a 3 log decrease in viral DNA and a rescue of thymic atrophy. But what we found is that compared to the vehicle control, treatment just during that first week with the antiviral or infection on day of life 7 resulted in decreased gastritis, as well as decreased autoantibodies, really suggesting that replication during that neonatal time frame in the first 7 days of life is necessary to develop this autoimmune disease. We next want to characterize which immune cells were really driving this phenotype. And we had to be a little bit careful about this because we know that any depletion of a specific cell early in life could impact the susceptibility to the virus and also just the natural course of infection. And so we used antibodies to deplete or block cells or cytokines starting at 6 weeks post-infection after the acute infection had resolved. We also used MUMT mice that lacked B cells and lacked them at the time of infection. And what we found is that compared to our control that developed gastritis at a very high penetrance, depletion of all alpha beta T cells, CD4 T cells, or blockade of IL-17 resulted in a reduction of gastritis. And we did not see this when we depleted CD8 T cells or CD20 positive B cells. But when we infected the mu-MT mice that lacked B cells, we actually did see a decrease in gastritis, suggesting that CD4 T cells as well as IL-17 are necessary for development of gastritis, but that B cells are only necessary early in life. So we next wanted to identify which cells were sufficient to induce disease. And to do this, we harvested splenocytes as well as cells from stomach draining lymph nodes and enriched them for CD4, CD8, or B cells and transferred them to nude mice. And what we found is that only transfer of the CD4 T cells from MRV infected mice induced disease. We also performed an experiment where we co-transferred the CD4 T effector cells with regulatory T cells. And we first did this with adult regulatory T cells and did not see a decrease in gastritis. But when we co-transferred with neonatal regulatory T cells, we did see a decrease in gastritis, suggesting that perhaps this neonatal regulatory T cell population has a unique impact on suppressing disease. The other major question is this phenotype specific to the roseolovirus, or is that something that can be seen after infection with another virus that is also thymotropic? And so naturally, we turned to another beta herpes virus, murine cytomegalovirus, and performed a similar infection and saw that there was a thymocyte atrophy or thymic atrophy as well as thymocyte depletion. And while this was a transient event similar to murine roseolovirus, we did not see induction of gastritis, suggesting that, at least amongst the beta herpes viruses, murine roseolovirus is having some unique impact on inducing autoimmunity. So we wanted to know how this is happening and what potential mechanism is driving disease. And there are really kind of two prevailing theories in the field for virus-induced autoimmunity. The first one I'm going to touch on is molecular mimicry. And to simplify this, the idea here is that an antigen-presenting cell will take up a virus and display viral antigen. And if that viral antigen is similar enough to a self-antigen, you get cross-reactivity of T and B cells, that self-antigen. And in the setting of a pro-inflammatory environment, autoimmunity. Now, when we were trying to understand the autoimmune disease after MRV infection, we actually did a couple of different screening approaches. And one of those that I'm showing here is we did an autoantibody screen. And so this was a microarray for different autoantigens. And what we found is that compared to the uninfected control or the antiviral treated control, the MRV infected mice developed this broad repertoire of autoantibodies. And while this doesn't completely rule out molecular mimicry, it is much less likely that this virus has this many antigens that cross-react with self-antigens.
Yeah, that's too many. It's so many.
It is. Like I said, it doesn't completely rule it out, and I have done searches to try to see if any of these line up with viral antigens on the protein level. And that's not the end all be all for this analysis, but it doesn't fit as well. Right. One thing I will point out for this group is we do see anti-insulin antibodies. And when we were doing our histology screening for autoimmunity, we actually do see that in these mice at a lower penetrance than the gastritis, but still occurring, is there seems to be this inflammatory infiltrate around and even into some of the pancreatic islets. This is something that we plan on exploring further to identify how prevalent this is in these mice after infection, as well as what this looks like over time.
Yeah, no, that is a fascinating result. And I would ask, whether if you have any, thoughts or did you see any data that suggested there was any kind of beta specific interaction or alpha specific interaction or both?
Yeah, it's a great question, and I don't have an answer for you yet. The other thing we really need to look at is whether or not MRV is infecting the pancreas. We have some initial data suggesting that it does not seem to be replicating in the pancreas, but in terms of identifying islet-specific T cells and antibodies and other antibodies that are associated with type 1 diabetes, we haven't looked into yet, but it's a really interesting finding. And
Yeah. Well, you've got an excellent islet biologist right there at WashU with Jeff Millman.
yes, of
I'm
course.
sure he could always be tapped for some input.
Yeah, and I think looking into this a little further, identifying over time whether these mice, um, you know, show symptoms, show signs of insulitis and pancreatic inflammation, and also getting ahead of myself to the future directions, but um, uh, if there are any stimuli that potentially can increase the penetrance after this initial hit after neonatal infection. So these are all, I think, really interesting questions
Yeah, I
beyond
mean,
the
yeah,
initial publication.
what you've really established here is a novel and exciting model system for a whole host of people to explore.
Right, exactly. So I'll come back to this just a little bit more at the end, but in trying to get back to the mechanism, we also considered another prevailing theory of virus-induced autoimmunity, which is this idea of bystander effect, which I'm lumping together with epitope spread. Um, the idea here is that after a tissue is infected, there's cell death and inflammation. Antigen-presenting cells take up those dying cells and debris and display them to T and B cells. And if you get just enough recognition and then maturation of that response in the setting of inflammation, you could develop autoimmunity. But as I mentioned, when we look for viral replication in the stomach, shown here at 7 days post-infection, but also shown here at 12 weeks post-infection when we see disease, we actually don't see the virus in the stomach. And we went a step further by using an antiviral to inhibit any potential ongoing replication of the virus in the stomach and did not see a reduction in gastritis, really suggesting that this idea that the virus is replicating in the stomach and inducing autoimmune disease, this is likely not the mechanism. And so we turn back towards the thymus, where we see that thymic atrophy, we see that depletion of thymocytes, and I'm going to just very simplistically go over this model of what's happening to developing T cells in the thymus. And the idea here that I'm going to focus on is that there are cells in the thymus, like medullary thymic epithelial cells or mTECs, that are expressing tissue-restricted antigens, genes that would normally be expressed in the pancreas or the lung or the liver. And there are transcription factors like AIR that drive this expression. And these antigens are then displayed to developing T cells. And if these developing T cells recognize this antigen too strongly, they either undergo apoptosis and are removed from the population to make sure that these autoreactive T cells don't escape. Some of these that have a stronger recognition can also be shunted into the regulatory T cell population. But disruption of this process can result in escape of autoreactive T cells, dysregulation of the regulatory T cell development, and downstream autoimmunity. So we wanted to explore some of these major players in what we call central tolerance.
This may be
Now
oh, here you go. You're going to answer my question right now. So go ahead.
There are a lot of examples of viruses infecting or impacting the thymus. I listed some of them here, but there's this whole list that is from this really great um, review. The interesting thing is, in none of these studies have they shown development of autoimmunity unrelated to ongoing viral replication. So we decided to look a little bit more at what's happening to some of these major players in central tolerance after infection. And one of the cells we looked at were these medullary thymic epithelial cells that are so important for training the developing T cells. And we do, in fact, see out to 4 weeks post-infection a decrease in the number of these cells. Interestingly, at 1 week post-infection, we do not see a decrease. But thymic dendritic cells that also are important for central tolerance, we do see a decrease as early as 1 week post-infection. But like the thymocytes, this resolves by 4 weeks post-infection. When we look at expression of that transcription factor AIR, we also see a decrease. And this starts at 1 week post-infection and continues out to 4 weeks post-infection. And functionally, when we look 1 week post-infection, we do see a decrease in expression of these tissue-restricted antigens. Shown here, a couple of examples, an antigen from the stomach, an antigen from the thyroid, as well as INS2. And we can rescue this expression with antiviral treatment. So with this in mind, what we really want to know next is, what is the tropism of this virus in the thymus? Which cells is it infecting? Is this impact due to direct viral infection, or to just the inflammation or other cells that are inducing damage in the thymus? And so we turned to single-cell RNA sequencing, and we decided to look at 6 days post-infection, shown here by flow cytometry, before we lose all those CD4 positive thymocytes. And you can see by the UMAP as well as the proportion of cells that we see a very similar pattern here in the single cell proportion as we do by flow cytometry. But when we look for expression of viral genes, really across the sequential gene expression that you see in herpes viruses, what we see is that the double positive, double negative, as well as single positive thymocytes, those developing T cells, are positive for viral transcripts. But we also see that the mTECs are positive for viral transcripts. So it seems that these cells are directly infected. And moreover, we see major transcriptional differences, including notch signaling that's important for the T cell development, genes that are involved in negative selection, as well as you'd expect, changes in interferon signal.
Can you, Tarin, can you
Yeah.
comment on whether or not these type of data have been seen in Coxsackie infection?
Yeah, that's a great
Thymus,
question. So
in the thymus itself.
yeah, so definitely Coxsackie virus can infect the thymus and can induce thymic atrophy. Um, there, there is some data that is similar to what we found in terms of a depletion of some of these cells. And Coxsackie virus has definitely been related to type 1 diabetes. And
Yes,
part
correct.
of the thinking with that research has been a direct impact of infection in the pancreas, perhaps. Um, and that may be the case. Potentially, this could be another virus that acts similar to MRV and induces some type of initial hit that impacts tolerance downstream. Uh, it could be related in both ways, because this is this can be more, as opposed to MRV. This can be a little bit more thought of as an acute infection with Coxsackie virus, whereas MRV is a chronic infection with periodic low-level reactivation.
Yes.
No one that I'm aware of has done these studies, although this is something that I have proposed to do, and to try to understand we don't see the same phenotype when we infect with MCMV, even in terms of development of autoimmunity, even though MCMV does infect the thymus. But
The one,
perhaps
the
there
one,
are.
yeah, no, the perhaps there are. And the one question, the one thing that's so interesting about your work is that this, the pattern of sort of like the infection, remission, and then a little bit, another a round of MCV and then remission. That does mirror what is now, kind of put forward as the model for type 1. It's a one hit,
Right.
a remission during the prodrome, another hit,
Yeah.
a remission. So it's really interesting that this mechanism does kind of, does have that a similar progression.
Yeah, and perhaps not all viruses impact similar pathways to MRV that induces autoimmunity, but there may be other viruses that do. And another big question, in our model here, we think MRV is infecting the thymus and really disrupting these major players in central tolerance, and this is resulting in escape of autoreactive T cells and perhaps um, change in the regulatory T cell population, and this is what's inducing autoimmunity. But we're going from a neonatal to adult time frame, and we showed in this paper that the autoimmunity doesn't really overtly develop until out here, more in this adult time frame. And so one of the questions that remains is what's happening in between, and are there specific factors that are increasing the likelihood over time that these mice will develop autoimmunity? The other thing, speaking to this group specifically, is autoimmune gastritis is definitely related to type 1 diabetes as well as Hashimoto's thyroiditis. And this is from a 2008 review of several different papers showing just the percentage of people with autoimmune gastritis that also have type 1 diabetes or thyroiditis or Graves' disease. And so I think there's good evidence in just the general population that these autoimmune diseases are related, but we also know that in air deficiency or APOSED, that these patients develop gastritis, they develop type 1 diabetes, they develop thyroiditis, um, as well as a whole host of autoantibodies and other autoimmune conditions. So, you know, this is unlike air deficiency. The impact of MRV is transient, but perhaps gives us an idea about a very important window in development and how a viral insult um, can have a long-term impact on tolerance and autoimmunity.
Yeah, no, this is a very, very important body of work that you guys are developing. And the whole idea of narrowing down the window of sort of first hit, second hit during the progress to type 1 is really, I think, where the meat lies, in the meat of the sandwich. So this is an exciting approach you guys have developed, and looks like there's a lot of work to be done now with the model.
There is. As a virologist, I'm really excited to do some of this virology work and, you know, identifying what potential genes are involved that from the virus perspective. But on the immunology side, as an immunologist, I think we have a really tractable model to start studying this. We have tools like antivirals. We're developing viral tools to try to really identify the mechanism. We also have really great immunologic tools to study this. So I think there's a lot to do, but for our initial studies, but also potential links with type 1 diabetes, I think there's a lot of exciting work to be done.
Absolutely. Are you hiring any postdocs?
I am hiring right now. The lab is opening. We're a small lab right now. I do just want to quickly thank Wayne Yokoyama here, who is an amazing immunologist, supported me as I did this work as a postdoc. I know there's an upcoming talk on natural killer cells and type 1 diabetes, and natural killer cells is really his avenue of research, but has allowed me to explore this really exciting topic, as well as several other collaborators, Jose Saenz and Michael Paley. So yeah, we are hiring. We're looking for people to join the lab, and there are lots of projects to take on.
It's fantastic. I cannot wait to see what you come up with next, your next paper.
Yes.
Exciting time.
Looking forward to it.
Yeah.
We're getting there for sure.
Thank you again for joining us. We just had we had a couple people on here that dropped, but I really appreciate your sharing with us today. And as you know, we have a lot of views and listens on our YouTube channel and our podcast. And so we're hoping that this word will get out and people will reach out to you directly.
Great. I really appreciate the opportunity.
It's mutual. Thanks again for all you're doing.
All right.
Have a great rest of your day. Bye-bye.
All right. Thanks. You too. Bye-bye.